Good evening, everyone. It's 6:05, so maybe we can start. I'll admit people as and when they come in. Can someone tell me if my screen is visible and if I'm audible?
Yes, ma'am.
Okay, thank you so much. So, good evening, everyone. My name is Aishi, and I'm your PMRF research scholar from the Department of Chemistry, IIT Bombay, and I am a TA for this NPTEL course, Applied Environmental Microbiology. And today is the fifth live session for this course, and we'll be discussing some of the sample problems that might help you solve the assignments from this course. So, we'll move to the first question. Yeah. So, which of the following statements is true? Statements is or are true. 2D gel electrophoresis separates the samples by the isoelectric points and charge in that order. 2D gel electrophoresis is a technique used for separation of proteins. Metabolomics analysis is the analysis of metabolites by separating them on basis of M by Z ratio. And comparative genomic analysis is predominantly used to identify the functional similarities in populations. Anyone? 1 second. I have to admit. Yeah. Yeah. Anyone would like to try this? Try answering this? Anyone?
Option 2.
Option 2 is the only option that's correct. Okay, 1 second. This is a multiple choice question, let me tell you that. So, do you think any other option is correct? Option 2 is correct, you're right. Any other option is correct? Okay, I'll explain. So, option 2 and 3 are the correct options. We'll discuss a little bit about 2D gel electrophoresis, and I'll also tell you why option 3 is also correct. So let's talk about this 2D gel electrophoresis. It's a very powerful tool for proteomics work, and it is used for separation and fractionation of complex protein mixtures from biological samples. So when you're extracting from biological samples, maybe from cells that you have grown till a particular time point, you'll have a lot of proteins in that complex sample. So this is a technique to separate those protein mixtures from biological samples, and this technique separates proteins depending on two different steps. The first one is called the isoelectric focusing, which separates basically, 1 second, I will add the laser. Anyway, isoelectric focusing, which separates proteins according to the isoelectric points. According to the isoelectric points. And the second step is the SDS polyacrylamide gel, SDS page electrophoresis, which is a very common technique, I think, used in all the molecular biology, chemical biology labs. And this separates proteins based on the molecular weights or the relative molecular weight. Thus, thousands of proteins can be separated, and the information about the isoelectric focusing and the molecular weights can be obtained. So there are several steps for successful 2D analysis. So as we know, the isoelectric point is that pH in a particular, if there's a pH range, it's that particular pH at which the protein does not have any charge. It comes in its zwitter ionic form. It does not carry any excess charge. So the isoelectric points of different proteins will be different because of the different side chains basically. So 1 second. So this technique utilizes both the isoelectric points of the proteins and also SDS page technique. So the sample preparation is quite complex. So this is another image. So this is isoelectric focusing, and proteins are differentiated based on their PI values, isoelectric point values, and then SDS page gel, where proteins are separated based on their size. And after running this electrophoresis, a protein of interest can be, if found, in two closely bands of similar size but different PI. So even proteins of similar size can be separated because they will definitely have different pi, and if there are two proteins which are very similar in the values of their pi, they can be separated on the basis of their molecular weight. So this is how the entire process occurs. So speaking of sample preparation, the native samples that are there, they need to be converted to a physicochemical state suitable for the first dimension separation, which is isoelectric focusing. That will keep the native charge and the molecular weight of the constituent proteins. So in order to get good results, appropriate sample preparation is very important. And there are various ways to prepare the samples because of the difference of the types and origins of proteins. Ideally, the process will result in the complete solubilization, disaggregation, denaturation, and reduction of the proteins in the sample. So the first dimension separation will be according to the pi of the proteins. So proteins are basically amphoteric molecules, and the positive, negative, or zero net charge that they carry depends on the pH of the surroundings. So the isoelectric point is basically defined as the pH of the solution at which the net charge of the protein becomes zero. So a protein with a positive net charge will migrate towards the cathode, becoming less positively charged until it reaches its pi, and at that point it becomes the net charge is zero. And a protein with a negative net charge will migrate towards the anode and become less negatively charged until it also reaches its pi. So to be specific, a protein mixture is loaded at the basic end of the pH gradient gel. So this is via pH gradient gel, and after applying electric field, the proteins are separated depending on the charges, focusing at the positions where the pi value is equivalent to the surrounding pH. And larger proteins will move more slowly through the gel, but with sufficient time will catch up with the small proteins of equal charge. Then this is the first dimension separation, after which there is the second dimension preparation that happens, which is on the vertical side. It is called the SDS page, which is an electrophoretic method for separating polypeptides according to their molecular weight. And this method often involves 4 steps, including preparation of the gel, the equilibration of the immobilized pH gradient strips in SDS buffer, placing the equilibrated IPG strip on the SDS gel, and then finally running the electrophoresis experiment. And SDS can make proteins denaturing, and basically SDS is a detergent. It is added to denature the proteins, and also to add SDS is added at a constant molar ratio to the backbone of the proteins. So when applying SDS and a reducing agent like DTT, it can cleave disulfide bonds. Proteins unfold into their linear chains with negative charge proportional to the polypeptide chain length. So SDS imparts a uniform negative charge to all the proteins depending on the polypeptide chain length. So eventually the separation will be based on molecular weight and chain length only. So polyacrylamide forms a mesh-like matrix which is appropriate for separating the proteins, and when proteins are separated by SDS page, smaller proteins obviously will migrate faster since there is less resistance. They can easily migrate faster because in the mesh. After that, there are various methods for visualization of the separated proteins. But the most commonly used is Kumasi blue staining or silver staining. Silver staining is a more sensitive and non-radio method, but the principle of silver staining is basically very simple. The amino acid side chains can bind to the silver ions, primarily the sulfhydryl and the carboxyl groups of proteins, followed by reduction to free metallic silver. As a result, the protein bands are visualized as spots where the reduction occurs. And silver staining is basically suitable for low protein levels because of its its sensitivity. It is sensitive in the low nanogram range also. So if you have a very low concentration of protein, silver staining is more suitable for visualization. Kumasi blue staining is the most common method that happens mostly, and it's a very relative simple method and more quantitative than silver staining. So it's usually used to detect protein bands containing 0.2 microgram or more proteins. And this Kumasi blue dye binds to proteins to form a protein dye complex to van der Waals interactions. And there are 2 types of Kumasi dyes like R250 and G250. So there are almost no possibilities to detect the appearance of a few new spots or the disappearance of a single spots in large studies with several thousand spots. And in addition, evaluation of 2 gels by manual comparison is also quite difficult and time consuming. Therefore, it is necessary to detect differences and obtain information from gels by image collection hardware, and there are quite a few image analysis software as well, and the gels can be used for the identification of other purposes also. So this 2D two differential 2D dimensional electrophoresis is a very widely used method for protein analysis with the ability to separate thousands of proteins at the same time, and it can actually provide sorry for interrupting the session so many times. Actually, I have to admit. And it can also provide direct visual information of changes in the PTM or the post-translational modification of proteins, and it can be also used for further analysis like proteome analysis, detection of biomarkers, drug discovery, etc. So all this happens, and basically, as I told you already, it's basically separated. Proteins will it's separated based on PI and SDA page. So to summarize, separation when it's happening, when IEF or isoelectric focusing is happening, a gradient of pH is applied to the gel, and the electric potential is applied across the gel, making one end more positive than the other, and at all pH values other than the isoelectric point, the proteins will be charged. And if, as I said, if they're positively charged, they'll be pulled to one side, negatively charged pulled to other side. So the proteins applied in the first dimension separation will move along the gel and will accumulate at their isoelectric points. That is the point at which the overall net charge in the protein is 0, and then separation will happen by molecular weight or SDA page. So at first the proteins will be denatured. It will unfold the proteins into long straight molecules. A number of SDA molecules will bind to it in a ratio that is relevant to the polypeptide chain length. And because a protein's length when it is unfolded is roughly proportional to its mass, SDS will impart a negative charge to the backbone, and as a result of this, all the proteins will have approximately the same mass to charge ratio as each other. And also, in addition, proteins will not migrate when they have no charge because of the isoelectric focusing step. So the coating of the protein in SDS will allow migration of the proteins in the second dimension. And at the second dimension, a electric potential is again applied, but at the 90-degree angle from the the first separation happens horizontally, the second separation will happen vertically, and the proteins will be attracted to the more positive side of the gel because, and the lower part is the positive, upper part is negative, and proteins will be attracted to the positive part because we know that SDS, we have exposed it to SDS, and SDS has actually imparted a uniform negative charge on all the proteins. So all the proteins are now uniformly negative charged based on their mass to charge ratio. So based on their mass, now they'll anyway move towards the positive end because of the SDS negative charge on the proteins, but this will happen according to the molecular weight. So this is the entire pattern. The sample preparation is quite complex for the first for both the dimension, but it's a very useful technique to separate a lot of proteins when we have a sample, biological sample like cells and all. So SDS page is a very common technique used in almost all the labs. 2D electrophoresis might be used in a very few labs, but SDS is very important. Yeah. So these are the techniques. So this is how the sample preparation. This is for the other option. So the other option that we said was true in this question was metabolomics analysis is the analysis of metabolites by separating them on the basis of mass to charge ratio. So this is also a correct statement. So the first statement is not correct because in this electrophoresis, 2D gel electrophoresis, the samples are separated by isoelectric points and mass, not charge. So this statement is wrong. Second option is correct, as she said. Third option is also correct. I'll explain why. Fourth option is again incorrect because comparative genomic analysis is not used to identify the functional similarities in protein. Functional similarities in population mostly arise from similarities in protein structures and all. So comparative genomic analysis might help in creation of a phylogenetic tree and all, but when we're talking about functional similarities, that is not quite corresponding to comparative genomic analysis. So fourth option is also not exactly correct. Third option is correct. So what is metabolomics? Metabolomics is basically the study of the metabolome within cells, biofluids, tissues, or even organisms to comprehensively identify and quantify all the endogenous and exogenous low molecular weight small molecules or metabolites in a biological system in a high throughput manner. And this metabolomics has several applications in health and disease, including precision or personalized medicine, single cell epidemiologic population studies, metabolic phenotyping, and also metabolome wide association studies. Then precision metabolomics, and in combination with other omics disciplines such as integrative omics, biotechnology, and bioengineering. Basically, proteomics means the proteome analysis, genomics means genome analysis. So metabolomics means basically the small molecule or metabolite analysis. And mass spectrometry or MS based metabolomics, it provides a very useful approach for both identification of disease related metabolites in biofluids or tissue, and also encompasses classification or characterization of disease or treatment associated molecular patterns generated from metabolites. So this is a brief schematic to show how it happens. uh, First, there is sample preparation and extraction. So when you're extracting samples, maybe from cell samples, or you have biological samples from hospitals, if you take, you have to harvest the samples, you have to control the environment at the time of harvesting, you have to keep the sample snap freeze in liquid nitrogen and keep it. Then you have to do enzyme quenching standards, then you have to spike the standards that you are using. Standards basically are some standard metabolites that you'll be using to compare if those metabolites are present in your cells or not, your samples or not. So this is like the protocol varies if you're taking a plant, if you're taking a tissue sample, if you're taking an in vivo sample or an in vitro sample. This protocol varies, but this is just a general schematic. So you'll take the sample, you'll extract, and this is then you'll move it to the mass spectrometry analysis. Mass spectrometry basically is used to identify and quantify metabolites even at very low concentrations, like even femtomolar, which is 10 to the power 15, I guess, to automolar, with high resolution, sensitivity, and dynamic range. And these MS based analysis basically include sample preparation, extraction, capillary electrophoresis or chromatographic separation, introduction of sample for ionization, and detection of possible metabolites on the basis of their mass to charge ratio. So this chromatography mass spectrometry is the last step. Once you perform the experiment, then you have to basically check in a database that has already been published or that is already there available, and you have to check which if the mass to charge ratio of the metabolites are actually matching with any metabolites that already exist in the database. So that's how it works. The extraction thing is very subjective to all the samples that you have. So you have to actually basically rely on a previously published paper, you have to follow the protocol, you have to optimize the entire thing. So the sample preparation extraction process is it depends on the samples that you have and what you're trying to check. But the mass spectrometry analysis is how it happens. So this entire process is called metabolomics. So the omics discipline has transcriptomics, proteomics, genomics, and metabolomics. So basically these, even when we talk about proteomics, we take a cell sample or whatever sample you have, we try to extract the proteins, and then we then we basically do mass spectrometric analysis of those proteins to check which proteins are present, and we do the mass spec analysis. Then we try to match the data with a previously published database to see if that protein is there in our sample or not. So that is why that option is also correct. Okay, so also comparative genomics basically was the third or fourth option, I guess. Yeah, so comparative genomic analysis basically it helps in identifying DNA sequences that have been conserved, that is preserved in many different organisms over millions of years, and it is an important step towards understanding the genome itself. It basically pinpoints genes that are essential to life and highlights genomic signals that control gene function across many species, and it helps us to further understand what genes relate to various biological systems, which in turn may translate into innovative approaches for treating human diseases and improving human health. And comparative genomics also provides a powerful tool for studying evolution. So by taking advantage of and analyzing the evolutionary relationships between the species and the corresponding differences in the DNA, scientists can better understand how the appearance, behavior, and the biology of living things has changed over time. But for functional analysis, comparative genomics is not exactly how it works out. So this is how the comparative genomics works. They are checking the conserved DNA in all the organisms. What are the common features of different organisms? They check a closely related species. It might help in showing a phylogenetic. It might help in creating a phylogenetic tree, because and also genetic differences might reveal variants with a rolling disease. That might also happen. Okay, so moving on to the next question. Which of these is most likely to be identical? Which of these is likely to be most identical? Orthologs or paralogs? Anybody? to answer? Okay, I'll go. It's a paradox. I'll tell you why. So this image basically illustrates the difference. So this part A of the diagram above shows a hypothetical evolutionary history of a gene. So the ancestral genome had two copies of this gene, A and B, which were paralogs. So the two copies of the genes are paralogs. At some point, the ancestral species split into two daughter species, as you can see from here. The 1 and 2 denotes the two daughter species, and each of whose genome contains two copies of the ancestral duplicated genes A1, A2 and B1, B2. So these genes are all homologous to one another, but are they paralogs or orthologs? So since the duplication event that created genes A and B occurred, one second, I will use the pointer. Since the duplicated event that created the genes A and B occurred before the speciation event. Speciation into daughter species happened after the duplication event. The created species 1 and 2 A genes will be paralogs of the B genes, and the 1 genes will be orthologs of the 2 genes. That means A1 and B1 are paralogs. A1 and B2 are paralogs. A2 and B1 are paralogs. A2, B2 are paralogs. But A1 and A2 are orthologs, and B1 and B2 are also orthologs. But this is a very simple case. What happens if the duplication occurs after the speciation event? So in this part B of the diagram, the ancestral gene was duplicated only in the species 2 lineage. So in that case, A2 and B2 are orthologs of A1, and A2 and B2 are paralogs of each other. So A2 and B2 are paralogs of each other, and because the speciation event has happened before the duplication event. So A2 and B2 are orthologs of each other, and A2 and B2 are orthologs of A1, and A2 and B2 are paralogs of each other. So that's why paralogs are most likely to be identical, because you can see these are A and B are also paralogs, because these are basically the two copies of a particular ancestral gene. So paralogs have more chances of being identical than orthologs. So difference between orthologous and paralogous genes. So orthologous genes are the homologous genes in different species due to separation by speciation. So it happens after the speciation has occurred, as you can see from the previous sketch as well. However, paralogous genes are the homologous genes found within a single species due to duplication. So this has happened A and B. There are two copies of the same. It happened in duplication. So that's why their paralogs are most similar to each other. Orthologs appear when like A1 and A2 are orthologs, because they are in two different species. So there is a speciation happening. That's what has led to the orthologs, right? A1 and A2 are orthologs, correct. And orthologous genes carry out the same function as the ancestral gene, but paralogous genes can have different functions other than the function of the ancestral gene. Orthologous genes appear due to speciation, paralogous genes appear due to genetic duplication event. Orthologous genes will belong to different species, very obvious, because it has emerged due to speciation. Paralogous genes will be within the genome of a single species. Orthologous genes are similar to the ancestral genes. We can see A and B will be very similar to the ancestral gene. Orthologous, paralogous is a combination of ancestral and new genes. So that is how this entire thing happens. So moving on to the next question. Choose the correct option. Rec A activates Lex A to find the error in DNA replication, and Lex A is error prone repair and hence regulated by Rec A. Which one do you think is true? Anybody? Sorry? Did you say something? Okay. I'm not sure if you said something I couldn't hear. But anyway, I'll answer. So both of these are not really true. Both 1 and 2 and false. So first we'll learn about what rec and lexa are, what these things are, and what is the SOS response in that happens. So the SOS response is a global response to DNA damage in which the cell cycle is arrested and DNA repair and mutagenesis is induced. So this system involves the REC-A protein. This is a REC-A protein, and it is in eukaryotes it is known as RAD51, but in prokaryotes it's a REC-A protein, and this REC-A protein is stimulated by single-stranded DNA involved in the inactivation, as this REC-A protein is involved in the inactivation of the repressor LEX-A of SOS response genes, thereby inducing the response. So it is an error-prone repair system that contributes significantly to DNA damage observed in a wide range of species. So what happens is that SOS response is initiated by the accumulation of single-stranded DNA during replication of DNA-containing lesions. So single-stranded DNA is generated when DNA polymerase, basically replication is happening, and DNA polymerase stalls at a template lesion when the replicative helicase continues unwinding DNA. So recent studies indicate that the replisome skipping over template lesions and repriming to allow reinitiation of DNA synthesis downstream of a block, leaving behind a single-stranded DNA gap, is possible during both leading and lagging strand synthesis. So it should be mentioned that during horizontal gene transfer, single-stranded DNA is also transiently present, and both transformation and conjugative plasmid DNA induce this SOS response. However, a number of conjugative plasmids encode a particular protein, which can even inhibit the induction of this SOS response during conjugation. So two proteins, REC-A and LEX-A, they play major roles in the regulation of the SOS response. This LEX-A protein is composed of two domains, basically two domains separated by a short flexible linker, the N-terminal DNA binding domain, which is called the NTD, and the C-terminal catalytic domain, which is called the CTD. And there's a serine with the serine lysine catalytic diet, basically. So the C-terminal domain is also responsible for the homodimerization of LEX-A. So during normal growth, this LEX-A dimer acts as a transcriptional repressor for genes belonging to the SOS regulon by binding to the specific operator sequence in the promoter region. But this REC-A acts as a co-protease to stimulate the self-cleavage of LEX-A as well as other related proteins such as Faj repressors, UMUD protein and its homologs. And this REC-A, what happens, it binds the single-stranded DNA, and in the presence of a nucleoside triphosphate converts to an activated form, which is the REC-A star, shown by the REC-A star. And this REC-A star stimulates the self-cleavage of LEX-A at a specific alanine 84 and glycine 85 bond near the middle of the protein, thereby deactivating LEX-A. So it deactivates LEX-A, it doesn't activate LEX-A. And this deactivation leads to lowering of its affinity for the DNA and exposing residues that target LEX-A for the LON protease degradation. And as a result, the pools of LEX-A protein begin to decrease, leading to depression of the derepression of the SOS genes. So this figure basically schematically shows this process, the SOS induction process in E. coli. So during normal growth, the LEX-A repressor binds to the operator sequence in the promoter region of SOS genes, preventing their expression. So upon DNA damage, single-stranded DNA accumulates, which basically leads to activation of REC-A, and when a replication fork stalls during replication of DNA, since REC-A binds the single-stranded DNA and in the presence of ATP converts to an activated form, which is the REC-A star. And this REC-A stimulates self-cleavage of LEX-A, leading to derepression of the SOS genes. And after the damage has been addressed, the elimination of the inducing signal allows LEX-A to reaccumulate and repress the SOS genes. So that's why the answer is both the options are false in this question, because LEX-A, it basically REC-A deactivates LEX-A to find the error in DNA replication. Okay, now moving on to the next question. This is a very basic question. Anybody? The taxonomical rank in order is anybody? What is the taxonomical rank?
False.
Anybody else? So you're correct. This is false because obviously it's not shown in the right order. So the right order of taxonomy in living things is domain, then kingdom, then phylum, class, order, family, genus, species. So for humans, Homo sapiens, domain will be eukarya, Kingdom will be Animalia, definitely not plants. Phylum will be Chordata. Class Mammalia. Order Primates. Family Hominidae. Genus Homo, and species Sapiens. So this is the correct order of taxonomy. What has been mentioned in this question is not correct. It's wrong. Great, she is right. So moving on to the next question. Several environmental species of hydrocarbon eating bacteria like Alcanivorous burcumensis feast on the spilled oil, assisting with disaster cleanup efforts. This is an example of anybody?
Bioremediation.
Correct. You're correct. It's bioremediation. So what is bioremediation basically? So and how does this process help? So with the increase in boating and offshore drilling and transportation of oil, all the coastal communities are in need of a plan basically to clean up waterways in the event of an offshore oil spill. And this manual removal of oil is a critical first step, and when paired with bioremediation as a secondary method, the highest possible success of oil breakdown at a spill site can be achieved. And unlike dispersants, bioremediation is a clean way to speed up the regeneration of oil in the waterways. And according to actually a research conducted by scientists in 1994, given that proper nutrients are present, an oil spill that was estimated to be cleaned by natural conditions in 5 to 10 years could actually be cleaned in 2 to 5 years with the use of bioremediation. So this particular microbe which has been mentioned in the question, Alcanivorous borcumensis, it's an oil degrading microbe, and it was basically added to water with a heavy fuel oil. And after 2 months, the water was clean enough to return back to the seas. And this bacteria is found in marine ecosystems and can absorb and digest linear and branched alkanes that are found in crude oil and its products. So, community resilience to oil spills would involve establishing a preparedness plan with a system in place to quickly implement these bioremediation methods. So, establishing oil spill preparedness practices in coastal communities will be critical to reduce the impact from oil, because the system would incorporate culturing tanks, quantities of hydrocarbon elastic bacteria in pellet form, and the means to transport and apply this in the field. So, and when oil spills occur, we know that they can cause major damage to the environment. There have been many oil spills in Alaska with devastating environmental effects, and many marine mammals have been killed. Fisheries have been closed, and native populations have lost their food source as a result of oil spills. And it is very important for communities to have a plan for what to do in the event of an oil spill. So the process of bioremediation does exactly this. It utilizes oil degrading bacteria to clean up the spill, and it has already been used in various oil spill instances, including the spill in the Prince William Sound. So adding bacteria nutrients to oil spills will significantly increase the degradation of the oil with less environmental impacts than other cleanup methods. And this will also help the communities to be more resilient and recover more quickly after the spill. So basically it uses the use of naturally occurring or introduced microorganisms or other forms of life to consume or break down this environmental population in order to clean up the polluted areas. This is safer for the environment than any other chemical or physical solutions, because the microorganisms involved with bioremediation are either already living in the affected environment or they are brought into the environment. And if the chosen bioremediation process involves the naturally occurring microorganisms in the ocean environment, then the group organizing the bioremediation cleanup would add 2 nutrients to the water to help boost population growth of the native species of bacteria. So this technique was used in Alaska during the Exxon Valdez oil spill in the Prince William Sound. Fertilizer was added to beaches in order to supply nutrients to the naturally occurring microbes in the water, and the oil in these fertilized areas biodegraded after about 2 times faster than the untreated controls. And the other main method of bioremediation is to take cultured microorganisms in lab and introduce them into an oil spill and then continue to feed them, making the population grow. So basically the composition of the crude oil is made up of 4 main elements. It usually contains 84 to 87% carbon, 11 to 14% hydrogen, 0.1 to 8% sulfur, 0.1 to 1.8% nitrogen, and 1 to 1.5% oxygen. So although there are also sodium, nitrogen and oxygen compounds in soil, the most common molecules are hydrocarbons. And there are 3 main groups of hydrocarbon molecules in the crude oil: the aromatics, the naphthalenes, and the alkanes. And the most difficult hydrocarbons for bacteria to biodegrade are the aromatic compounds. because aromatic compounds are double bonded, they're carbon rings, and some of them are also attached to chains of hydrocarbons. So very small aromatics evaporate of a spill and or can be biodegraded. Larger aromatics, however, resist biodegradation and can persist in the area of a spill for a really long time, killing the marine ecosystem. And the only way that they can be broken down is by photooxidation or degradation by UV light. So aromatics are also one of the most toxic components in the crude oil, like naphthalene. Naphthalene are single bonded saturated hydrocarbon rings. Naphthalenes can be biodegraded more easily than aromatics, but not as quickly as alkenes because they contain more bonds. But at least naphthalenes are single bonded circular structures, so it's easier to biodegrade than aromatics, which are double bonded structures. Alkanes are easier to biodegrade because they are straight or branch saturated hydrocarbons. They only contain single bonds, which is ideal for microbial degradation because it does not take much energy to break apart the molecules compared to double bonded molecules. They can be solids, liquids, or gases depending on the number of carbon atoms they contain. And alkanes with one to like alkanes with 1 to 4 carbon atoms are gases, so they're also volatile compounds. So during an oil spill, these compounds evaporate off the slick and into the air. But alkanes with 5 to 16 carbon atoms are liquids, and these form most of the oil sleek. They can be degraded relatively quickly by bacteria. The smaller the chain, the easier it is for the bacteria to break it down. But alkanes with more than 16 carbon atoms are solids and are very difficult for bacteria to break down. So the characteristics of this crude oil, this also differ from place to place. Like it has been shown that the Alaskan North Slope crude oil has a relatively high viscosity and forms an emulsion with water very quickly, and this makes it extremely important to respond quickly to spills made up of this type of oil. So with the exception of aromatic compounds, all hydrocarbons in crude oil can be easily degraded by bacteria, and this makes bioremediation a very important method for cleaning up oil spills. And in time, a naturally occurring bacteria will completely break down a spill. However, they cannot do this quickly enough to prevent damage to the ecosystem unless bioremediation methods are used to speed up the growth. So this Alcanivorax borcumensis is that marine bacteria that can absorb and digest this linear and branched alkanes that are found in this crude oil and its products. And it's basically a gram-negative bacteria. Basically, that means that the bacteria has an outer membrane of lipopolysaccharides, unlike gram-positive bacteria, which do not possess this layer. And it is also rod-shaped bacteria that is aerobic, that is, it depends on oxygen, it is oxygen reliant. And it is basically included in the genus Bacillus, which is a genus for rod-shaped bacteria. That is in the class gamma proteobacteria, meaning that it's scientifically important bacteria. And since this particular bacteria already occurs naturally in unpolluted waters all over the world, including fresh water, it has to have a source of energy. And there's a particular study that has found that the strains of two of the most abundant cyanobacteria in the ocean, namely Proclococcus and Cyanococcus, they produce and accumulate hydrocarbons, particularly alkanes C15 and C17. And these alkanes are the energy source for Alcanivorax borcumensis in the unpolluted water. So this particular bacteria, Alcanivorax borcumensis, it naturally flourishes after an oil spill because there is more abundant source of energy. It also participates. This abundant source of energy actually helps to sustain a larger population, and this bacteria also participates in the wastewater treatment by being formed by Nocardia species. And this bacteria also breaks apart the bonds in hydrocarbons in oil that have been exposed to the sea using enzymes and oxygen found in the seawater. So I think it creates enzymes named as the ALKB1 and ALKB2. ALKB1 is involved in the direct reversal of alkylation damage, specifically in the single-stranded DNA, and ALKB1 hydroxylases alkanes with 8 to 16 carbons, and the chain lengths with the most 5. And they have the alkanosis growth, Alcanivorax borcumensis growth are actually 14 to 19 carbon chains dependent, and is able to outcompete other hydrocarbon-elastic species of bacteria because it can break down such a wide range of alkane chains. But this bacteria cannot consume sugars or amino acids, source of energy, unlike most other bacteria. And in terms of the biology, the cell biology, it basically is a bacterium. It contains a single ringed chromosome free floating in the cytoplasm, and this single stranded circular chromosome contains islands. It's these islands that code for the bacteria's oil degrading enzymes. The islands are made up of genes that code for different enzymes, and these genes are mobile, meaning that they can change their position in the chromosome, also allowing the islands to transfer to other bacteria of different species through horizontal gene transfer. In this way, also non-oil degrading microbes can develop the enzyme creation by horizontal gene transfer with this Alkinivorax borkumensis. Then they'll also be able to degrade oil. So that is the speciality of this particular bacteria. So this is known as bioremediation, and if you are interested in learning more about Alkinivorax borkumensis, you can go and actually do a Google search. Whatever information I said are actually from like rigorous Google search only. I got this information from a paper that has actually conducted quite a lot of study on this particular bacteria. So moving on to the next question. Plants get infected by viroid by both acquiring the infection from infected neighboring plants via air and by coming in contact with the infected cells. It's true or false? Anyone?
True.
Okay, you think it's true. I think it's false. I'll explain why. So, virions are infectious agents that consist only of naked RNA without any protective layer such as a protein core, and they infect plant only, no other forms of life, and are replicated at the expense of the host cell. And virio genomes are small single-stranded circles of RNA that are only 250 to 400 bases long. So, and as we know that viruses all encode at least one protein needed for the replication of the virus genome, but virioid RNA does not contain any gene that encode proteins. It merely carries signal for its own replication by the host machinery. So, although the viriod encodes no protein enzymes, the virioid RNA itself acts as a ribozyme. That is, the RNA catalyzes an enzymatic reaction. And whether or not the viriod has any genes depends on whether we count the sequence of RNA that persists ribozyme activity as a gene. So virioids have no protein coding genes, but the virioid RNA itself acts as a ribozyme. And these virioids replicate by a rolling circle mechanism, and the virioid's own ribozyme activity is used for self-cleavage of the multimeric RNA generated during the replication, and the host enzymes provides all other functions. The first host RNA polymerase copies the circular plus strand to form a multimeric minus strand. Then site-specific cleavage of this strand by the virioid ribozyme gives monomers that are circularized by a host RNA ligase. And the minus stranded circles are the templates for a second round of rolling circle replication by RNA polymerase, and the resulting multimeric plus strand undergoes ribozyme cleavage to create monomers, and these are circularized to produce the progeny virioids, which are circular positive single-stranded RNA. Single-stranded RNA. So most virioids replicate in the plant cell nucleus and rely on RNA polymerase 2 for RNA synthesis. And a smaller group of virioids, such as the chrysanthemum chlorotic motile virioid, they have a highly branched structure rather than a rod with budges and replicate in the chloroplast. So some virioids cause no detectable symptoms in their host plants, whereas others cause massive damage. So how virioids damage plants is still rather mysterious. There are studies going on. However, it appears that the virioid and its replicative intermediates trigger RNA interference, and this results in the formation of short single-stranded RNA molecules known as virioid short RNA of the same size as the microRNAs widely used by plant cells to regulate genes, and this decreases expression of multiple plant genes, causing various diseases. So virioids basically cause plant disease by triggering RNA interference. So the question was, by both acquiring infected neighboring plants via air and by coming in contact with the infected cells, air is definitely not exactly true. Virioids mostly infect plants by triggering any kind of RNA interference that is happening, because the RNA interference which will have a massive regulation on the genes that are needed for the plants to survive, and if it is decreasing the expression of essential genes, then there will be diseases occurring in the plants. So that is why virioids are such fascinating elements, and studies are still going on to understand the exact mechanism that virioids use to infect plants. Okay, moving on to the next question. Match the following: Archaebacteria protozoa and eukaryotic, ubiquitous, present almost everywhere, associated with extreme conditions. Which one do you think is the correct answer? Anyone?
Option 3.
Option 3. 1 C, 2 B, 3A. Correct. So, archaea, bacteria and archaea are microbes consisting of only a single cell. They are similar in many aspects, including like size, shape, reproduction, habitat. Both are much simpler than any eukaryotic cell and are considered to be the early forms of life on Earth. However, they also have many differences that eventually separate them into two biological domains. And basically, bacteria are single cell microbes with cell walls. They are found almost everywhere on Earth. They are found on everywhere, and that's why almost ubiquity is present almost everywhere. Some live or on other organisms, including plants and animals as well. The human body itself is full of bacteria. Many of these bacterial cells are found living in the gut and are important for our health. That we need that microbiome of the gut for our health. However, some bacteria are also pathogenic. It can cause bacterial infections, illness, and disease. What are archaea? Archaea are another group of single cell microbes. They were originally classified as ancient bacteria, but later they received their own domain. Archaea are so different from the bacteria that they must have a long independent evolutionary history since close to the dawn of life. And some archaea live in extreme environmental conditions. That's very difficult for any other life forms to survive. Some archaea can survive high temperatures, like often above 100 degrees Celsius, as found in geysers or hydrothermal vents and oil wells. Others can live in extremely chilly, icy habitats, highly saline or acidic or alkaline water, and this unique ability gives them the name of extremophiles. So, what do bacteria and archaea have in common? Both of them are prokaryotes. They do not have any kind of membrane bound nucleus. The genetic element DNA is within the cytoplasm. They also lack any other membrane bound cell organelles. They don't have mitochondria or chloroplast or endoplasmic reticulum or Golgi body, etc. For this reason, all the biochemical reactions as well as transcription translation happen directly in cytoplasm. And bacterial and archaeal cells cannot grow big also due to this limitation. Also, both bacteria and archaea are simple single cellular organisms. Each bacterial and archaeal cell is an independent organism. They can't form organized tissues or organs as multicellular organisms like us. For this reason, each bacterial and archaeal cell has to do everything that a living organism needs, feeding and reproduction. And both archaea and bacteria like to form a community. So, although bacteria and archaea are single cells, they tend to grow together into a high density community of cells called biofilms. And living in a biofilm gives the individual cells many advantages. For example, the matrix of biofilm protects the cells with high resistance from any kind of antibiotic penetration. Also, bacteria and archaea have similar sizes and shapes. They're pretty tiny, almost in the range of 1 to 1.5 micron. Although there are some exceptions, some bacteria can actually grow up to even 750 micron, while nanoarchaeota can be as small as zero point 0.4 micron as well. Also, bacteria and archaea are both found as rods, cocci, spirals, plates, or coil. They both have hair-like appendages called cilia, called flagella, and pili or pili on their cell surfaces. For this reason, it is challenged. It is very challenging to distinguish between bacteria and archaea by just looking at their morphology. Also, both bacteria and archaea are found everywhere on Earth. They are very successful in terms of their adaptability. You can find them everywhere. The ocean has the most abundant amounts of bacteria and archaea. They both have members who can survive under extreme conditions, including hot springs, deep sea vents, saline pools, icy oceans. So that's why they are called extremophiles, and they're also abundant in our bodies. And both bacteria and archaea can use diverse energy sources. So when the ancestral life forms evolved, Earth's early environment may not be as friendly as it is today. And bacteria and archaea had the diversity. They had to diversify the energy sources in order to expand the territory. In both the groups, there are phototrophs that obtain energy directly from sunlight. There are chemotrophs that rely on energy from certain chemical reactions, and there are organotrophs which live by breaking down organic materials. But there are also a number of critical differences between bacteria and archaea, and these differences are so substantial that actually archaea they are in a different domain. The cell type of bacteria is different. Both of them are prokaryote. Cell morphology might be quite similar, but archaea lack peptidoglycan. They are made of protein, whereas bacterial cell wall is made of peptidoglycan. Then, in case of bacteria, the cell membrane composition is of ester linked with lipids with D-glycerol straight chain. But in case of archaea, there are ether linked lipids with L-glycerol branch chain. Both will have 78 ribosomes. Both will have flagella. Archaeal flagella are also called archaea. Both will have pili. The chromosomal DNA for both is long circular form. The plasmid both can have plasmids as well. Then there are some unique structures in case of bacteria called capsule. In case of archaea, they are called cannulae or hammers. Then in bacteria, some can be pathogens. That's not that kind of that popular in case of archaea. Some bacterial species can do photosynthesis like cyanobacteria. In archaea, some species are phototrophs, but they do not perform such oxygen generating photosynthesis. Bacteria can use glycolysis or Krebs cycle. Archaea generally do not use any such thing. Both habitat wise, archaea are more diverse. They can live, they are basically the extremophiles. They are like, this term is basically for archaea only. They can live in deep sea, various diverse environments. Some bacteria can also live in such environments. So that is why this happens. The main difference is that bacterial cell wall contains peptidoglycan, but the archaeal cell wall, so peptidoglycan is composed of polysaccharide chains and peptides, and these cell walls can be thinner in gram-negative bacteria than gram-positive bacteria, because gram-positive bacteria also have lipoteichoic acids which anchor the cell wall to the cell membrane. And some pathogenic bacteria have an extra thick layer of sugars outside the cell wall, which is called capsule. But the archaeal cells do not have peptidoglycan. The protection is provided by the surface layer proteins called the S layer, and archaea do not have any kind of capsule. Archaeal plasma membrane are made of molecules that are different from those in other life forms. So, in most organisms like bacteria or eukaryotes, the cell membranes are made of molecules known as phospholipids, which have a phosphate head group and a lipid tail. And the lipid fatty acid in the membranes of other organisms have straight carbon chains. However, the archaeal lipid tails are long isoprenoid chains with multiple side branches and rings. And these branch chains may prevent archaeal membranes from leaking at high temperatures. Another unique feature of the archaeal membrane is that it is composed of glycerol ether lipids, whereas bacteria and eukaryotes have membranes composed of mainly of glycerol ester lipids. In case of flagella and pili, both bacteria and archaea have hair-like structures on their cells. The long one is called flagella, while the short one is called pilus. So this is what we've already discussed. There's also another column talking about eukarya. So this basically table sums up the differences between these three domains. So this matching, that's why archaea will match with associated with extreme environments, bacteria will match with ubiquitous, present almost everywhere, and protozoa is eukaryote. So this is how protozoa falls in the proteus group, and it's basically a eukaryote. Okay, moving on to the next question. Algae are photosynthetic eukaryotes. Anybody? True or false?
False.
You think it's false? But some algae can do photosynthesis. It's there in this example. Algae is a eukaryote. Some algae can do photosynthesis. So I think it's true. And the unusual biology of these algae arose from endosymbiosis. I've already discussed the process of endosymbiosis when we're talking about mitochondria. So what happens is, basically endosymbiosis when a cell lives in an irreversible symbiosis within another cell. So endosymbiosis is it's a very important concept, because according to some scientists, the photosynthetic organelles such as plastids of all photosynthetic eukaryotes ultimately trace back to a cyanobacterium, which is a prokaryote that took up residence within a eukaryotic host more than billion years ago. And basically eukaryotes are cells, organisms with nucleated cells such as animals, plants, fungi, whereas cyanobacteria are prokaryotes whose cells do not have nucleus or any other membrane bound organelles. And this fateful encounter is called primary endobiosis because it led to all the plastids known today. So during an evolutionary event referred to as the primary endosymbiosis, an eukaryotic cell irreversibly assimilated a prokaryotic cell called a cyanobacterium, and this generated three lineages of algae. There is the Glaucophyta, the Rhodophyta, and the Chlorophyta that contain a photosynthetic organelle, plastid, which is a remnant of that cyanobacterium. And which is a remnant of that cyanobacterium, and mitochondria, which are organelles derived from the integration of other prokaryotes early in the eukaryotic evolution. So members of one of these lineages, the chlorophytes, gave rise to all the extant complex and land plants. So, in two independent evolutionary events, other non-photosynthetic eukaryotic hosts then engulfed cells from both the rhodophyta, which is the red algae, and the chlorophyta, which is the green algae lineages. And this secondary wave of endosymbiosis led to two further algal groups, the cryptophytes and the chloroarachinophytes. And these unicellular organisms contain not only plastids and mitochondria, but also an organelle called the nucleomorph, which is the relict of the nucleus of the engulfed algae. So that's why it's said that algae are eukaryotes which can do photosynthesis, because they must have engulfed a cyanobacterium in a primary endosymbiosis event. We have discussed endosymbiosis and we're discussing about the unique genetic element of mitochondria, as we know that mitochondria has a unique genetic element of their own. But how is that possible? Because it's an organelle. So this theory says that mitochondria used to be an independent organism before, prokaryotic organism before, which was engulfed by a eukaryotic cell, leading, and then finally it evolved into a mitochondria. So this endosymbiosis theory also supports this explanation of the emergence of photosynthetic eukaryotes, which is algae. So this is true. Moving on to the next question. With regard to the PRNP gene discussed in the lecture, PRP-SC misfolded proteins are regarded as pathogens because this mutates genetic coding of the cell. True or false? Anybody? Anybody has any idea about the prion disease, how these things happen? Okay, so this is false. PRP, SP, SC misfolded proteins are indeed pathogen. They have the pathogenic ability, but not because it mutates the genetic coding of the cell, for a completely different reason. So the prion disease is a collection of fatal transmissible neurodegenerative diseases that cause rapid onset dementia and ultimately death. Uniquely, the infectious agent is not a bacteria or a virus. The infectious agent is a misfolded form of the endogenous cellular prion protein termed as PRPSC. So, despite the identity of the molecular agent remaining the same, PRPSC can cause a range of diseases with hereditary, spontaneous, or various other, complexities. So, however, the link between PRPSC and toxicity is complex, with subclinical cases of prion disease discovered and prion neurodegeneration without obvious PRPC deposition. So, the toxic mechanisms by which PRPSC causes the extensive neuropathology are still studied. They are still poorly understood. Although recent advances are beginning to unravel the molecular underpinnings, including oxidative stress, disruption of proteostasis, and induction of the unfolded protein response. So there are multiple reviews online you can search which discusses the disease caused by this PRPSC toxicity, the nature of the toxicity of PRPSC, and our current understanding of the downstream toxic signaling events triggered by the presence of PRPSC. So what happens is that it's basically this range of neurodegenerative disorders are characterized by extensive neuronal death, spongiform change, and gliosis. So uniquely among these neurodegenerative diseases, the prion disease are transmissible with infection between members of the same species, and in some cases between different species also. And since the discovery that the infectious component of prions is composed solely of protein, more specifically a misfolded form of the cellular prion protein, a great deal of research has to focus on how this misfolded protein can cause such extensive and catastrophic damage to neurons. The major histopathological feature is the accumulation of extracellular amyloid plaques comprised of misfolded PRPC. However, it is still not known known properly how PRPSC causes neurodegeneration, or indeed what the exact toxic species is, as the link between the PRPSC infectivity and toxicity is not sharply defined, and the role of this PRPSC is also elusive, further clouding the investigation into this. So the PRPC is the wild type protein. It's the human prion protein. It's highly conserved in mammals. Wait one second. It's highly conserved in mammals, suggesting an essential role for the protein. And it's a small glycoprotein found mainly on the outer leaflet of the plasma membrane, held in place by a C-terminal glycosyl phosphatidylinositol or GPI anchor. So although highly expressed in the tissues of the central nervous system, PRPC is also expressed to varying degrees in most tissues in the body. And expression begins early in the embryogenesis and in adults The highest levels are in neurons, with moderate expression observed in glial cells and the peripheral nervous system. And human PC is synthesized as a 231 amino acid polypeptide um, after removal of a 22 residue signal peptide, and this is processed through the endoplasmic reticulum and Golgi apparatus. There are post-translational modifications, including the removal of a signal sequence from the C-terminal end of PRPC, resulting in a mature protein of 208 amino acids in length. And the main structural features are a globular C-terminal domain made up of 3 alpha helices with a small antiparallel beta sheet composed of 2 separate strands and a largely unstructured flexible N-terminal tail. And the exact cellular function of this PRPC remains unclear, with several distinct and overlapping roles suggested. One of the most important proposed functions of this PRPC is the maintenance of myelination in the peripheral nervous system. Interestingly, neuron-specific PRPC expression is enough to maintain myelination, as although PRPC is expressed in Schwann cells, it appears to be not essential there. And it is unknown what effect PRPC has on the central nervous system myelin. And the first proposed role for PRPC was in copper homeostasis due to its high affinity binding. Although the functional physiological role for this affinity still remains elusive. And this PRPC has a putative role in protecting against stress, especially oxidative and some apoptotic stresses. And there is also evidence it helps to regulate neuronal excitability and memory. Interestingly, it's also involved in the regulation of the circadian rhythm and cellular differentiation. The wide variety of these biological roles has led to the suggestion that PRPC is a scaffold protein that regulates the formation of a number of multi-protein complexes, but it is unlikely that the entirety of its physiological roles have been discovered. So this is the wild-type protein, not the misfolded one. The crucial event in the development of this prion disease is the structural and conformational change of this PRPC to the disease-associated structure called the PRPSC. It's basically a misfolded protein. This conversion changes PRPC from a protein characterized by alpha helices to a partially protease resistant misfolded protein characterized by beta sheets. Proteinase K can partially digest this misfolded PRPSC and is often used to determine the presence of misfolded PRPSC. Despite this conversion being essential for the pathogenesis of prion disease, the molecular mechanism is still under study. And the transformation is thought to be a post-translational change in conformation, which initiates the catalytic conversion of PRPC into more PRPSC by the interaction of existing PRPSC molecules. So existing PRPSC molecules will get recruited, and it will also change the correctly folded PRPC into PRPSC. And this continuous synthesis of PRPC in the brain only provides more substrate for the pathological conversion to PRPSC. And while this mainly occurs after exposure to already misfolded PRPSC, conversion can occur spontaneously in rare cases also without exposure or a genetic basis. And there are no primary sequence differences between the PRPC and PRPSC. So the change is mediated by different secondary structures and a propensity to aggregate. And this pathological change involving only the prion protein is summarized in the protein-only hypothesis. Strong evidence supporting PRPSC as the main cause of prion disease comes from the production of the infectious PSC in vitro. Whatever the mechanism, this conversion is the basis of all the prion disease. So the misfolding happens, PRPSC gets formed, and more PRPSC gets recruited, and it changes the normal wild type PRPC protein into more and more misfolded proteins, and these are very protease resistant, so these cannot be degraded also easily, and that ultimately leads to the majority of the observed neurodegeneration, because there is aggregation of this PRPSC that happens, and it appears to be toxic, leading to more and more. Importantly, PRPC is actually required for both the conversion process and the manifestation of the toxicity of PRPSC. So that's why PRPSC misfolded proteins are very pathogenic, but it doesn't mutate the genetic coding of the cell. Genetic coding of the cell doesn't get mutated, but it actually gets recruited to change the correctly folded wild type PRPC proteins into more and more misfolded proteins. That's why it's so pathogenic. Moving on to the next question. Which factor allows the rhizobial cells to invade the root cells in legumes? Anyone?
Node factor.
Correct. So node factor is the correct answer. So basically, we know that the bacteria in the genera Rhizobium, Bradyribosium, and Azoribosium, they collectively form the rhizobia, and they form complex symbiotic relationships with legume roots in the form of nitrogen fixing nodules. And this association is established in a host specific and guest specific manner that involves a two-way exchange of chemical signals. And the first signal comes from the host plant in the form of a mixture of flavonoids released from the legume root, and this signal induces the synthesis by receptive nodulating bacteria of signal molecules known as the Nod factors. So these Nod factors cause the legume root cells to become mitotically active and to differentiate into nodule structures. And these factors are composed of chitin oligosaccharide backbones modified with a variety of substituents and are synthesized by enzymes encoded by the bacterial nodulation genes. And these nodulation genes have been classified as either common or host specific. A nodulation gene is considered common if it can be complemented by a gene from another species without altering the host range of the recipient bacteria. For example, Rhizobium meliloti forms nodules with the host plant alpha alpha, but not with the vetch. And the converse is true of Rhizobium leguminosarum biovar vici. So Rhizobium meliloti with a mutation of a common nodulation gene can be complemented to form nodules on alpha alpha by cloned DNA from Rhizobium leguminosarum uh, vici. So these ribosium meliloti transconjugates cannot nodulate with, so their host range was unchanged. The same theme has been found across quite diverse species of Rhizobium, Bradyribosium, and Azoribosium. So this host specific nodulation gene, on the other hand, cannot functionally complement basically mutations across species and often alter the host range of the recipient rhizobium when transferred from one species to another. So the common nodulation genes encode enzymes that catalyze the synthesis of the Nod factor code. Specifically, the Nod C protein is a chitin oligosaccharide synthase that polymerizes UDP-N acetyl D-glucosamine into chitin tetraose and chitin pentaose. And the Nod B protein basically is known to deacetylate the terminal N-acetyl glucosamide at the non-reducing end of the chitin oligosaccharides. And the Nod A protein is an acyl transferase that adds an N-linked acyl substituent to the deacylated chitin oligosaccharide. And the identity of the N-linked acyl moiety is determined by the host specific genes Nod EF, the products of which assist in the synthesis of special polyunsaturated fatty acids. So yes, the answer was correct. It's a Nod factor. So this is how the nodulation thing happens. So the leguminous plant releases the flavonoids. As I said, this is a two-way chemical exchange process. The leguminous plants will release flavonoids into the area surrounding the plant roots, known as the rhizosphere, where the available nitrogen in the soil is depleted. Then rhizobia in the surrounding soil will secrete Nod D, which is a protein that recognizes the flavonoids secreted by the plants. Then there will be interaction with the flavonoids, which will activate Nod D. Then Nod D will return to the rhizobium to induce Nod D will return to the rhizobium, which will finally induce the transcription of the Nod ABC genes. The Nod ABC proteins that are formed will modify the Nod factor in response to the flavonoids released by the plant. And the Nod factors will interact with the plant and cause root hair curling and root hair deformation, and this will lead to the formation of an infection thread in the root hair. Nod factors cause cortical cell division in the primordium of the plant and form a nodule, as you can see here, and the rhizobium moves through the plant root hair by the infection thread to the nodule within the plant. So this is the entire schematic of nodule development. So it's a Nod factor, correct. So this is another schematic to show how the Nod factors affect this entire thing. The flavonoids are released by the plant, and the Nod factors are the complementary corresponding structures from the bacteria. So at first, the interaction of the flavonoids with the Nod D protein determines if the nodulation genes are induced. This is the first step in determining the host range. Then the Nod factor is synthesized by the nodulation gene products. Then the Nod factor will induce the legume root to form a nodule. The specific structure of the Nod factor determines its activity on various hosts. Then the rhizobia will invade the root to complete formation of the symbiotic nitrogen fixing nodule. Moving on to the next question. Where's my this question? This question. When a bacteriophage infects the host cell, which of the following will enter the host? Outer part of virion, protein coat, capsid made up of capsomeres, host genetic material, all of the above, none of the above. Anyone? anyone knows how the answer will be, you can try.
Option
All of the
2,
above.
3.
2, 3. Someone else was also
Yes.
saying.
All of the above.
It's actually none of the above. Uh, we'll explain what bacteriophage is and what are the two cycles that bacteriophages follow. So the answer will be none of the above. So I'll explain. So bacteriophage basically are viruses for the bacteria. So the viruses that infect bacteria are called bacteriophage. And there are two different cycles that the bacteriophages may use to infect their bacterial hosts: the lytic cell cycle and the lysogenic cycle. So in the lytic cycle, the phage will infect the bacteria, hijack the bacteria to make lots of phages, and then kill the bacteria by lysing it, basically, like making it explode. In the lysogenic cycle, the phage will infect the bacteria. It will insert its DNA into the bacterial chromosome, allowing the phage DNA, now which is after the phage DNA is incorporated into the host cell, it is called prophage, and it will be copied and passed on along the with the cell's own DNA. So there was no option of the phage DNA. Basically, there's host genetic material, but that's not what happens. Had there been a phage genetic material, phage thing there, that would have been the correct option. None of these things are entering the host. So what happens in the so this is a schematic of a bacteria, very rough schematic. It infects bacteria. Bacteriophages can also vary a lot in their shape and genetic materials. It can consist of either DNA or RNA and can contain as few as 4 genes or even 100, several hundred. And the capsid of the bacteriophage can be icosahedral, filamentous, or head tail in shape. And the head tail structures seem to be unique to phages and their close relatives. And these are not found in eukaryotic viruses. Viruses which infect, eukaryotic cells. So what happens? This is the lytic cycle schematic. So what happens in the lytic cycle? As you know, bacteriophages, just like other viruses, must infect a host cell in order to reproduce. The steps that make up the infection process are collectively called the life cycle. So some phages can only reproduce via a lytic life cycle, in which they burst and kill the host cells. Other phages can alternate between a lytic life cycle and a lysogenic life cycle, in which they don't kill the host cell as such and are instead copied along with the host DNA each time the cell divides. Basically, their genome gets integrated into the host cell genome. So let's take a closer look at both these cycles. As an example, we'll use a phage called lambda, which infects E. coli bacteria and can switch between the lytic and the lysogenic cycle. So what happens in the lytic cycle? A phage acts like a typical virus. It hijacks the host cell and uses the cell's resources to make lots of new phages, causing the cell to lyse and burst and die in the process. So you can see the phage is infecting the bacteria. This is the bacterial chromosome. The phage is phage is basically the phage DNA, phage genetic element is entering the bacteria. It's not the covering, not the outer capsid or anything. The phage DNA enters the bacteria. Then this phage DNA is getting replicated and protein synthesis is happening, and all of this is happening using the resources and everything from the host bacteria. So basically, the phage DNA hijacks the host cellular machinery to replicate its own DNA. Then, as their DNA replication is happening, protein synthesis or capsid protein synthesis also occurred. So the newly assembled phage also get created in the bacterial cell, and after that the bacterial cell, the phage basically lyses, makes the bacterial cell explode, and it comes out of the bacteria. So the first step will be the attachment step, in which the proteins in the tail of the phage binds to a specific receptor. It can be any sugar transporter or anything on the surface of the bacterial cell. Then the second stage is the entry. The phage will inject its double-stranded DNA genome into the cytoplasm of the bacteria. The third will be then DNA copying and protein synthesis. The phage DNA is getting copied and phage genes are expressed to make the proteins, such as the capsid proteins. Then there is assembly of the new phage. So now the DNA replication has occurred and protein synthesis also occurred. So capsids will assemble from the capsid proteins and are stuffed with DNA to make lots of new phage particles. Then lysis will happen. Late in the lytic cycle, the phage will express genes for proteins that poke holes in the plasma membrane and cell wall, and the holes let water flow in, making the cell expand and burst like an overfilled, water balloon. So cells bursting or lysis that happens releases hundreds of new phages in the environment, which can find and infect other host cells that are nearby. So these are the stages of the lytic cycle. Then moving on to the lysogenic cycle. So the lysogenic cycle doesn't burst the cell, it basically allows the phage to reproduce without killing its host. Kind of like a parasitic interaction. Some phages can only use the lytic cycle, but the phage that we are describing, the lambda phage, which basically infects E. coli bacteria, it can switch between the two cycles. So in the lysogenic cycle, the first two steps, the attachment and the DNA injection, occur just similarly, just as they do for the lytic cycle. Like it's very similar. However, once the phage DNA is inside the cell, it is not immediately copied or expressed to make proteins. It thinks of the long run. So what it does, it recombines in the particular region of the bacterial chromosome, and this causes the phage DNA to be integrated into the bacterial chromosome. So the phage DNA is now recombined with the bacterial chromosome, and it is now integrated into the bacterial chromosome. Now it's called the prophage. So similarly, attachment also happens how it was described previously, and entry also happens, and but the DNA is now incorporated into the bacterial chromosome. So each time a cell containing this prophage divides, its daughter cells will inherit the bacteriophage DNA, which is called the prophage. And the integrated phage DNA, called the prophage, is not really active. Its genes aren't expressed, and it doesn't drive production of new phages. However, each time a host cell divides, the prophage is copied along with the host DNA, getting a free ride basically. And the lysogenic cycle is less flashy than the lytic cycle, but at the end of the day, it's just another way for the phage to maintain its reproduce and maintain its existence basically. So under the right conditions, the prophage can become active and come out of the bacterial chromosome, triggering the remaining steps of the lytic cycle basically, like DNA copying and protein synthesis, capsid assembly, phage assembly, and lysis. So how does basically a phage decide whether to enter the lytic or the lysogenic cycle when it infects a bacterium? So one important factor is the number of phages infecting the cell at once. Larger numbers of co-infecting phages make it more likely that the infection will use the lysogenic cycle, and this strategy may help prevent the phages from wiping out the bacterial hosts by toning down the attack if the phage to host ratio gets too high. And what triggers a prophage to pop back out of the chromosome and enter the lytic cycle? At least in the lab, DNA damaging agents like UV radiation and chemicals will trigger most prophages to in a population to deactivate. However, a small fraction of the prophages in a population spontaneously go lytic even without these external triggers, external cues. So before antibiotics were discovered, there was considerable research on bacteriophages as a treatment for human bacterial diseases. So you can see bacteria can infect humans, and if you want to kill the bacteria, we want a virus that will infect the bacteria. So bacteriophage is like enemy's enemy is a friend, that kind of thing. The enemy of humans will be the bacteria which is causing the infection, and the virus or the bacteriophage that is infecting the bacteria will be a friend of humans. So before the entire concept of antibiotics was discovered or studied, people actually try to see if this bacteriophage can be used to treat human bacterial diseases, because bacteriophages attack only host bacteria, it will not attack the human cells. So they are potentially very good candidates to treat bacterial diseases in humans. But after antibiotics were discovered, the phage approach was largely abandoned in many parts of the world. However, phages continue to be used for medical purposes in a number of countries, including Russia, Georgia, Poland, where they still remain in use today. And there's an increasing interest in bringing back this phage approach elsewhere as antibiotic resistant bacteria become more and more a problem. We know that people generally don't consult the doctor. They one second. There are more and more multi-drug resistant or antibiotic resistant bacteria that are coming into existence. There are multiple drug resistant mycobacterium, tuberculosis, and most of this happens because people don't consult the doctors, proper qualified people for taking these antibiotics. They think that antibiotics will help anything and everything. So even with a mild cold, people are taking antibiotics these days without any proper consultation with the doctor, and this is making more and more bacteria resistant to antibiotics. So there are more and more antibiotic resistant bacteria that are developing, and it's a very huge crisis. So in some parts of the world, this phage concept is actually like being looked into more vigorously, so that if there are antibiotic resistant bacteria coming up, then if this concept be used to help solve this problem, because bacteriophage won't affect us. So can they be used to treat bacterial cells? So this more and more research is happening. in today's time. So that is why this bacteriophage versus antibiotics is a very important topic. And first, the other critical significance that, like as I said, many highly pathogenic bacterial toxins are encoded by bacteriophage genomes, such that the host bacterium is only pathogenic when lysogenized by the toxin encoding phage. Examples are cholera toxin in Vibrio cholerae, diphtheria toxin in Corynebacterium diphtheria, botulinum neurotoxin in Clostridium botulinum. All this Shiga toxin of the Shigella species. So without their phage encoded toxins, these bacterial species are either much less pathogenic or not pathogenic at all. So why phages encode these toxins is still not known. So while cholera toxin arguably helps both the phage and its host reach the next victim by inducing copious watery diarrhea, the paralysis resulting from the botulinum toxin would seem to have the opposite effect. Second, also bacteriophages are very good vectors for horizontal gene transfer, including antimicrobial resistance genes. They also have been engineered to introduce genes into specific strains for clinical effect, although this use is currently in the testing stage. And a third clinically very relevant use aspect of bacteriophages is that their detection can be used as a biomarker for the presence of their host in a complex environmental sample. So bacteriophages will only exist in their host. So if bacteriophage can serve as a biomarker, so if bacteriophage presence is detected in a sample, that means their host is also present. And this is most commonly is used as a surrogate for fecal contamination of water resources. So if the phage is present, the host most likely is present as well. And alternatively, these phages have been engineered to produce detectable molecules such as the luciferase. So the phages are genetically engineered in the lab so that their DNA includes the gene for this molecule, luciferase. So when they infect their host as a means to detect bacteria, they can actually light up, and that will help detect their host in a mixed environmental sample. And while mostly supplanted by newer technologies, bacteriophages also are clinically very relevant for their ability to distinguish strains of the same bacterial species. So most species of bacteria studied have multiple bacteriophage pathogens, just as humans as a species are susceptible to multiple viruses. So different species within a particular different strains within a particular species are resistant to some phages and not others. So by infecting each strain systematically with a standardized panel of phages for that species, each strain can be identified by the pattern of susceptibility and resistance to each phage type. So phage typing of Staphylococcus aureus, for example, utilized a standardized panel of bacteriophages shared internationally to differentiate various strains of Staphylococcus aureus. And before the development of molecular methods for this purpose, such multi-locus sequence typing and pulse field electrophoresis phage typing was the criteria standard for tracking strains for epidemiological purposes. And also finally, bacteriophages were the first type of virus to be discovered and were a part of the many of the fundamental discoveries of molecular biology. For example, the proof that DNA was the molecule that transmitted genetic information, the basic mechanism of gene regulation, and genetic code to name, but few were all discovered using bacteriophages. And as I talked about the antibiotic resistance already, like bacteriophages are drivers of bacterial evolution in the human microbiome. So prophage can be induced to switch to the lytic replication cycle by host cell stress, including antimicrobial drugs. So therefore, antibiotic treatments, especially those targeting gut microbial flora, can be expected to alter the phage microbiome as well. And bacteriophages are not as easily inactivated as vegetative bacterial cells, but they are vulnerable to UV inactivation, autoclaving, and standard hospital disinfection techniques. So that's why bacteriophage is still very much clinically relevant. Moving on to the next question: which of the following is true with regards to coronavirus disease 2019 or COVID-19 basically? Anybody? Is coronavirus a DNA virus or an RNA virus? Anybody has that knowledge? So it's a positive stranded, like plus as a single stranded RNA virus. It's also an enveloped virus. So viruses, as we know, can be differentiated based on how they store the genetic information, such as in DNA or double stranded RNA. And positive sense single-stranded RNA viruses are one such where it's a key aspect of the infectious cycle of the virus. Two important examples of positive stranded ssRNA viruses are the severe acute respiratory syndrome coronavirus 2 or the SARS-CoV-2 and hepatovirus A, which causes coronavirus and hepatitis A respectively. Hepatovirus A causes hepatitis A. So, how do these positive single-stranded RNA genomes replicate? It occurs in the cytoplasm of the host cell and often occurs in tandem with the assembly of the nucleocapsid into which the genetic material is packaged. However, there are some exceptions to this, such as the enterovirus, and this differs from the replication of double-stranded DNA and RNA viruses, where empty capsids are made first and later filled by the genome. And the close relationship between replication of the viral genome and nucleocapsid formation means that the genomic RNA and the capsid protein are highly specific for each other. Despite this, the positive single-stranded RNA viruses are a diverse category of viruses, and RNA replication and packaging specificity can differ between even closely related viruses. In many cases, the replication of positive single-stranded RNA viruses occurs spontaneously, without ATP and thus no energetic cost. And in vitro experiments have also shown that positive single-stranded RNA viral assembly depends on interactions between proteins and between proteins and RNA. The genomes are the templates for translation and replication, which is what fosters interactions between the host replication factors and RNA replication at several levels. So the positive sense RNA viruses make up more than one-third of all known virus genera, and this includes important pathogens, but also many viruses on official lists of potential bioterrorism agents. And these types of viruses use host factors in actually all steps of viral infection, such as entry, replication, etc. And most importantly, positive strand single-stranded viruses, RNA viruses, can modulate the gene expression and defenses of the host by co-opting host factors. So the genome RNA of a single-stranded RNA virus contains only the genes needed for the infectious cycle and is simultaneously a messenger RNA. Its packaging depends on several different segments, which is believed to be an adaptation to withstand mutations. And given the close relationship between RNA and proteins, a mutation could interrupt the infectious cycle if the packaging relied on a limited number of genes. And host cells typically they have certain defense mechanisms against these type of viruses. Endogenous deaminases have been known to edit the RNA and DNA of pathogens to defend the host against the pathogen. In mammals, adenosine deaminases acting on RNAs and apolipoprotein B mRNA editing enzymes also exist. So these act on RNAs and DNAs where they deaminate adenines into inosines and cytosines into uracils for and adenosine deaminases and apolipoprotein B, etc. So, like all other coronaviruses, SARS-CoV-2 is also a positive single-stranded RNA virus. And while much study remains to be done on COVID-19, there are some signs that human host molecules attempt to restrict the spread of the virus by editing their RNA through the use of adenosine deaminases and apolipoprotein B. And the few studies done have found low levels of editing, but some have found that parts of the cellular transcripts have been highly edited as well. And the full effect of this editing and the nature of the positive single-stranded RNA virus genome is still unclear. But single-stranded RNA viruses are very compact and compare only the necessary genetic information, which can be detrimental for the virus if there is editing occurring. However, while editing can cause the demise of a virus, it can also aid in its evolution. So yes, SARS-CoV-2 or coronavirus disease is a single-stranded RNA virus. This is a spike glycoprotein that basically attaches to the host cell ACE2 receptor, triggering the entry of the virus into the host cell. These are the membrane protein, the small envelope proteins, and it's covered by an envelope as well. There's a lipid envelope covering the virus as well. Yes. So moving on to the next question, luciferase gene enzyme essential for the pyrosequencing technique. Anyone?
True.
Yes, correct. So pyrosequencing is basically a method of DNA sequencing that detects light emitted during the sequential addition of nucleotides during the synthesis of a complementary strand of DNA. And it can be used to detect DNA and RNA from almost any source and has applications in bacteria resistance screening, drug development, epigenetics, and many other fields. So the steps, there are 6 major steps to pyrosequencing. The DNA that is to be sequenced is broken up into fragments of around 100 base pairs of single-stranded DNA. Then a polymerase chain reaction or a PCR is run to create millions of identical copies of each DNA fragment, which are split across thousands of wells. with just one type of DNA fragment per well. Then the DNA fragments are incubated with DNA polymerase, ATP sulfurylase, and apyrase enzymes, adenosine 5' phosphosulfate, and luciferin substrates. And one of the four types of nucleotides that make up DNA are added to the wells, which begin to be incorporated into the single-strand DNA template by DNA polymerase at the 3' end, releasing pyrophosphate. And ATP sulfurylase then converts pyrophosphate to adenosine triphosphate in the presence of adenosine 5' phosphosulfate. And ATP then takes part in the luciferase mediated conversion of luciferin to oxyluciferin. And this process emits light proportionately to the amount of ATP taking part in the conversion, which is picked up by a detector. And unused nucleotides in ATP degrade to apyrase, allowing the reaction to start again with another nucleotide. And this process is repeated, adding each nucleotide one after the other until the synthesis is completed. And a detector picks up the intensity of light emitted by the process, which is then used to infer the number and type of nucleotides added. For example, if 3 cytosine nucleotides are added sequentially to the same fragment of DNA, the light emitted will be 3 times more intense than the fragments of DNA with only one cytosine nucleotide added. So yes, the answer is true. It does involve luciferase enzyme. Next question. The FASTQ file carries the quality information along with sequences, while the FASTA file carries only the sequences. This is a little bit on the edges of bioinformatics. So is it true or false? Anybody?
True.
Okay, so it's true. So we'll talk a little bit about the differences between the FASTA and the FASTQ file. So the FASTA is basically a text-based format that only stores nucleotide or protein sequences, while FASTQ is a text-based format that stores both sequence and associated sequence quality values. FASTA is basically an alignment way software for DNA and protein sequences. It uses this FASTA format. It's a text-based format that represents either nucleotide sequences or amino acid sequences for protein, and here single letter codes represent both these sequences. And FASTA is a very important tool in the fields of bioinformatics, so it's a very common term. This format allows for sequence names and comments to precede the sequences, and this format originated from the FASTA software, which was introduced by David J. Lipman and William R. Pearson in 1980s. The FASTA tool had many modifications over time, and the latest version consists of programs for protein, DNA, protein translated DNA, and ordered or unordered peptide searches. The FASTA reads a given nucleotide or amino acid sequence and looks for the corresponding sequence database by using local sequence alignment to find matches of similar database sequences. What is FASTQ though? FASTQ is an alignment software used in the field of bioinformatics which stores both a biological sequence, usually a nucleotide sequence, and its corresponding quality scores. So FASTQ was originally developed to basically bundle a FASTA formatted sequence and the related quality data by the Wellcome Trust Sanger Institute. And with the development in the field of bioinformatics, FASTQ became the de facto standard for storing the output of many high throughput sequencing instruments. So the FASTQ format uses 4 different lines per sequence. Line 1 begins with @ character and is followed by a sequence identifier similar to a FASTA line title line. And line 2 consists of raw sequence letters. In line 3, the sequence begins with a plus character and is optionally followed by the same sequence identifier, and line 4 I think encodes the quality values for the sequence in the line 2 and should consist of the values of the same number of symbols as letters in the sequence. So what are the similarities between these? So these are both alignment tools. They are two sequence representation formats. They're also related to the field of bioinformatics. Both FASTA and FASTQ are important tools for storage and sequencing purposes. FASTQ is an extension of the FASTA format basically, with the ability to store the sequence quality as well. But what is the difference between FASTA and FASTQ? FASTA is a text-based format, only stores the nucleotide and protein sequences, while FASTQ FASTQ basically stores the quality scores as well. And this table summarizes the entire thing, difference between FASTA and FASTQ. So yes, the answer was correct. It's true. That's it. Transcriptomics is the metagenomics investigation of all messenger RNA. Anybody, it's true or false?
True.
Yeah. So when we talk about this omics platform, transcriptomics, we're talking about the mRNA transcripts, and we have already talked about metagenomics in one of the previous classes. So, what metagenomics is the study of the entire genetic content of all microbiota members in a natural habitat by utilization of the whole genome sequencing technique. Uh, and this field of metagenomics centers upon direct genetic analysis of microbial genomes isolated from various environments ranging from the human gastrointestinal tract to geothermal hot springs as well. It's an alternative approach to targeted amplicon sequencing in the study of uncultured microbiota. And by offering direct access to the entire genetic makeup of microbial communities, metagenomics can actually provide very valuable molecular insights into the novel enzymes and biocatalysts, as well as into the genomic linkages between community function and structure. So its metagenomics approach is a powerful tool for elucidating the relationship between host associated microbial communities and host phenotype. So that's why metagenomic sequencing is very important. What is transcriptome and transcriptomics? So transcriptome is basically the total RNA content of an organism or tissue type. It represents the various varieties of DNA and their copies in an organism or tissue. It's basically this omics format is responsible for active changes seen during the life cycle of an organism, and the total content of RNA is varies depending on the cell function or environmental conditions. At any given time, there could be thousands of RNA transcripts in an organism that represent the gene expression. And the study of RNA's content and sum total of RNA transcripts is called transcriptomics. So mRNA represents the protein content or the level of gene expression, while non-coding RNA do not code any proteins but perform the diverse functions. So this is a transcriptomic analysis schematic, and the transcriptomic analysis has propelled the ability to profile or characterize individual transcripts of the whole transcriptome. And transcriptomic analysis can be divided into two areas: the de novo transcriptome analysis, that is, in the absence of a known set of mRNA transcript draft, a transcript assembly is performed to characterize all mRNAs and isoforms in a transcriptome. The de novo transcriptome assembly is a complex process due to the alternative splicing events across the various tissues. And there is another gene expression profiling, wherein scientists understand the gene regulation in an organism or specific tissue under given environmental conditions. So true. Moving on to the next question. In horizontal transfer of a gene, conjugation, transformation, and transduction have to happen simultaneously. Anybody?
false,
False.
Yeah, it's false. There's no threshold like it has to happen, no threshold or no criteria that all the three things have to happen simultaneously. So conjugation, transformation, and transduction are three types of three ways to horizontally transfer a gene. So in transformation, the bacterium takes up a piece of DNA floating in its environment. In transduction, DNA is accidentally moved from one bacteria to another by a virus, and in conjugation, like DNA is transferred between bacteria through a tube called cells. So what happens in transformation? A bacteria takes in DNA from its environment, often DNA that's been shed by other bacteria. In a laboratory, however, we introduce DNA. Scientists introduce DNA, and if the DNA is a form of a circular DNA called plasmid, it can be copied in the receiving cells and passed on to descendants. So there are some bacteria which are competent, and competent cells can easily take in plasmid DNA, and we can actually genetically engineer the plasmid to incorporate our gene of interest. And if we finally transform the bacteria with the plasmid, we can basically enhance the production of that particular protein encoded by that gene. So why is this important? Imagine that a harmless bacteria takes up DNA for a toxin gene from a pathogenic species of bacteria, and if the receiving cell incorporates the new DNA into its chromosome, which can happen by the process of homologous recombination, it may also become pathogenic. So in normal cases, not transformation in labs. The next instance is transduction. In transduction, the viruses, we already talked about bacteriophages, which are viruses that infect bacteria. So viruses that infect bacteria move short pieces of chromosomal DNA from one bacteria to another, and when even bacteria can get a virus basically. And bacteriophages, they commandeer a cell's resources and use them to make more and more bacteriophages. And sometimes chunks of host cell DNA can get caught inside the new bacteriophage as they are made. When one of this is defective, the bacteria infects the cell, it transfers the DNA. Some bacteriophages even chop the DNA of their host cell into pieces, making their transfer more more likely. And as we already talked about the lytic and the lysogenic cycle, archaea, which is the another group of prokaryotes besides bacteria, they are not infected by bacterioviruses as such, but have their own viruses that move genetic material from one individual to another. Next is conjugation. In conjugation, the DNA is transferred from one bacterium to another after the donor cells, using tubes basically. So after the donor cell pulls itself close to the recipient using a structure called the pilus, DNA is transferred between the cells. In most cases, this DNA is in the form of a plasmid. And an F plus donor cell contains its chromosomal DNA and a F plasmid. Basically, it's a F plus donor cell. It has a rod-like pilus. A recipient F minus cell has only the chromosome and no F plasmid. The donor cell then uses its pilus to attach to the recipient cell, and the two cells are pulled together, and the channel or tube forms between the cytoplasm of the two cells, and a single strand of the F plasmid is fed through. And both the cells now have F plasmid and an F positive. The former recipient cell is now a new donor and can form a pilus. So donor cells typically act as donors because they have a chunk of DNA called the fertility factor or F factor, and this chunk of DNA codes for the proteins that make up the sex pilus, this one. And it also contains a special site where DNA transfer during conjugation begins. If the F factor is transferred during conjugation, the receiving cell turns into F plus or F positive structure, and then transfer DNA to other cells. So that's how it works. So these are the three types of genetic element transfer, how horizontal gene transfer is occurring. MGE means mobile genetic elements. So this is taken from a current opinion in microbiology journal. So this is how the three techniques help in horizontal gene transfer. So there is no written rule or no criteria that all three have to happen simultaneously. So they can happen individually as well. So that's why this answer is false. The answer to this question is false. So let's see. Genotypical differences may or may not lead to phenotypical differences. Anyone? It's true or false?
False.
Okay. Anyone else? So you think genotypical differences will always lead to phenotypical differences? So you're saying that it's true. Okay. So you're saying the genotypical differences will not always lead to phenotypical differences. So the answer is actually true. Genotypical differences may or may not lead to phenotypical differences. Before that, we'll talk about the difference between genotype and phenotype. So what is a genotype? A genotype and phenotype may sound very similar, but there's a huge difference between these two terms. The genotype is basically a set of genes in the DNA responsible for the unique traits or the characteristics, while the phenotype is the physical appearance or the characteristic of an organism. The human genetic code could be found by the genotype. It determines the traits which will be expressed. Organisms that look the same do not have the same genotype. Biological tests can determine genotypes, can make or might not have the same genotype. However, phenotype, as discussed already, both genotype and phenotype might sound similar. The phenotype is determined by an individual's genotype and expressed genes or by visible traits, for instance, hair color or type, color of eye, body shape, height. It depends on the genotype, but is also influenced by environmental factors. So these are the major differences. The hereditary information or the gene set is what forms a genotype, where in case of phenotype, it's basically the physical appearance that are visible to eyes. The same genotype produces the same phenotype. If everything is identical, that will lead to the same phenotype. The same phenotype may or may not belong to the same genotype. But if the entire genotype is same, if the genotype for blue colored eyes, everything is same, then both the children will be blue colored. But two random people who have blue colored eyes, blue color, just an example. Let's talk about brown hair. Two random people with brown hair might or might not have may belong to the same genotype. Genotype is present inside the body as genetic element. Phenotype is the expression, the physical appearance, external appearance. Genotype is obviously inherited from the parent to the offspring. The phenotype is not inherited. It's a function of it's dependent on the genotype, definitely. Genotype can be determined by various molecular biology methods like PCR, but phenotype can just be seen by observing the organism. Genotype is affected by genes, phenotype is affected by genotype and also environmental conditions. Genotype are blood group, eye color, height, and genetic diseases, and phenotype is weight, physique, beak or birth. So this example pretty much shows why this answer is true. So so we all know the concept of dominant and recessive. So in this case, these are the two alleles of this particular gene. The gene is for the flower color, and the two alleles are capital B and small b. Capital B is the dominant allele, and small b is the recessive allele. So when both dominant alleles are present in an organism, that is homozygously it's dominant, the color will be purple. When both small b's are present in an organism, that is homozygous recessive, it will be white, because small b represents white, capital B represents purple, and capital B is dominant over small b. But when capital B and small b are present together, that's the heterozygous population, that will be purple. That will be purple because capital B is dominant over the recessive allele small b. So that's why that will be purple. So the question was genotypical differences may or may not lead to phenotypical differences. So that's why it's true. So these two are genotypically different. Capital B, small b. The homozygous dominant and the heterozygous, these two are genotypically very different. One has two capital B's, one has one capital B, small b. So genotypically they are different, but they are not leading to any phenotypical differences. Both the flowers are purple in color, and that's because capital B is dominant over small b. That is the capital B is a dominant allele over the small b, which is a recessive allele. So this is a very beautiful example to show why this statement is true. So genotypical differences may or may not lead. There's no written rule that will always lead to phenotypical differences. So that's the way to explain this question. Moving on to the next question. Mutations always interfere with the normal transcription process. Anyone?
False.
Okay. So I also think it's false, and the way I interpret the question is that if there are mutations in random genes, the transcription process will occur, but obviously the mutated form of the protein will form. If there's a protein, a wild type protein, which is normally what is standardized available in the surroundings environment, and there's a mutation occurs, and the wild type protein changes to a different format. So that the transcription will not be hindered. Transcription will happen, but obviously a mutated form of the protein will be formed. So it is not exactly interfering with the normal transcription process. The process is going on, just that the normal protein is not formed. A different protein is formed. A mutated protein is formed. But sometimes when mutations are in specific regions, uh, such as the place where the transcription process will start, such as the place from where, like the various elements in the DNA which help in the transcription process, normal functioning transcription process. Like it's a quite a complex process. There are various factors, various proteins which come and attach to DNA and then help it get transformed to RNA. That's what transcription is, right? DNA to RNA. So there are many various protein factors that come and attached at various places of the DNA. So if mutations happen at those places, then that might lead that those kind of mutations might interfere with the transcription process. Might be the polymerase is not coming and attaching properly, or any other enzyme which is needed for the process to happen is not attaching properly because there is a random mutation in one of the sites where the enzyme will come and attach. That those kind of mutations will always interfere with the normal transcription process. So mutations in general, the word always is included in the question. That's why I feel it's not always interfering with the normal transcription process. So we know that there are different processes of mutations because DNA is constantly changing through the process of mutation. I'll discuss the process of mutation in a very brief manner because we know that DNA is dynamic and it's a very adaptable molecule. It's also stable, and as such the nucleotide sequence is found is subject to change in the change resulting to like leading to mutation, and depending on how a particular mutation modifies an organism's genetic makeup, it can prove harmless, helpful, or even like very damaging to the organism. Sometimes a mutation may even cause dramatic changes in the physiology of an affected organism. So it's necessary to understand what are the basic types of mutation and how they occur. So the first type of mutation which can occur, firstly there are basically mutations can be categorized into two categories, basically somatic mutations and germline mutations. Somatic mutations take place in non-reproductive cells. Many kinds of somatic mutations have no obvious effect on an organism because genetically normal body cells are able to compensate for the mutated cells. Nonetheless, certain other mutations can greatly impact the life and function of an organism. For example, somatic mutations that affect cell division, particularly those that allow cells to divide uncontrollably, are the basis for many forms of cancer. Germline mutations, which is the other category of mutation, occur in gametes or in cells that eventually produce gametes. So, in contrast with the somatic mutations, germline mutations are passed on to the organism's progeny as hereditary. As a result, future generations of organisms will also carry the same mutation in all of their cells, both somatic and germline. And what kind of mutations exist? Mutations aren't just grouped according to where they occur. Frequently, they are also categorized by the length of the nucleotide sequences that they affect. And changes to short stretches of nucleotides are called gene level mutations because these mutations affect the specific genes that provide instructions for various functional molecules, including proteins. Changes in these molecules can have an impact on any number of an organism's physical characteristics. As opposed to gene level mutations, mutations that alter longer stretches of DNA, ranging from multiple genes to an entire chromosome, are called chromosomal mutations. And these mutations often have serious consequences for affected organisms. Because gene level mutations are more common than chromosomal mutations, the sections that we are going to discuss focus on these smaller alterations to the normal genetic sequence alteration. So the first type is base substitution, which is shown here. So this is the simplest type of gene level mutation, and they involve the swapping of only one nucleotide for another during DNA replication. For example, during replication, a thymine nucleotide might be inserted in the place of a guanine nucleotide. And with base substitution mutations, only a single nucleotide within a gene sequence is changed. So only one codon is affected, as we can see from the sketch over here. It's not so base substitution is very the basic kind of mutation. Then we can come to insertion and deletion. So although base substitution alters only a single codon in the gene, it can still have a significant effect on protein production. In fact, depending on the nature of the codon change, base substitutions can lead to three different subcategories of mutations. The first of these subcategories can be missense mutations, in which the altered codon leads to the insertion of an incorrect amino acid into a protein molecule during translation. The second category can be nonsense mutation, in which the altered codon prematurely terminates synthesis of a protein molecule. And the third is the silent mutation, in which the altered codon codes for the exact same amino acid as the unaltered codon. So these are the three subcategories of base substitution mutation. The next is the insertion deletion. So insertion deletions are the two other types of mutations that can affect cells at the gene level. An insertion nucleotide occurs when an extra nucleotide is added to the DNA strand during replication, and this can happen when the replication strand slips or wrinkles, which allows the extra nucleotide to be incorporated. Strand slippage can also lead to deletion mutations. The other is deletion mutation, in which a wrinkle forms of the DNA template strand and subsequently causes a nucleotide to be omitted from the replicated strand. The final type of mutation that we'll discuss is the frameshift mutation. So insertion or deletion of one or more nucleotides during replication can also lead to this type of mutation called the frameshift mutation, and the outcome of this frameshift mutation is complete alteration of the amino acid sequence of a protein. And this alteration occurs during translation because ribosomes read the mRNA strand in terms of codons or groups of three nucleotides, and these groups are called the reading frames. So if the number of bases removed from or inserted into a segment of DNA is not a multiple of 3, the reading frame transcribed to the mRNA will be completely changed. And consequently, once it encounters the mutation, the ribosome will read the mRNA sequence differently, which can result in the production of an entirely different sequence of amino acids in the growing polypeptide chain. So to better understand, we can actually do this with an example. So as you can see the example over here. So this is with this triplet code, the sequence shown in the figure 5, it corresponds to a protein made of the following amino acids. Now suppose if a mutation occurs during replication and it results in the deletion of the fourth nucleotide in the sequence. So the fourth nucleotide is deleted, that is this particular A is deleted. So instead of the 3 A's, now there are 2 A's. So this particular nucleotide is deleted, and this is so when separated into triplet codons, the nucleotide sequence will now read like this. If the fourth nucleotide is deleted, so this will lead to a completely different set of amino acids. See, it was methionine, lysine, leucine, arginine, methionine previously. Now it's methionine, asparagine, alanine, glycine, and then stop. So it is getting prematurely terminated. The amino acid sequence is also completely different. So each of the stop codon tells the ribosome to terminate protein synthesis at that exact point. So consequently, the mutant protein is entirely different due to the deletion of this fourth nucleotide. It is also shorter due to the appearance of a premature short stop codon. So this mutant protein will obviously be unable to perform the function of the wild type protein. Now, what causes these kind of mutations? Mutations can arise in cells of all types. This is a variety of factors, including chance also. There is a chance spontaneous mutation as well. In fact, some of the mutations discussed here are a result of spontaneous events during replication, and thus they are known as spontaneous mutations. The slippage of the DNA template strand and subsequent insertion of an extra nucleotide is one example of a spontaneous mutation. Excess flexibility of the DNA strand and the subsequent mispairing of bases is another. Environmental exposure to certain chemicals, ultraviolet radiation, or any other external factors can also cause DNA to change. So these external agents of genetic change are also known as mutagens. And exposure to these kind of mutagens often causes alterations in the molecular structure of nucleotides, ultimately causing substitutions, insertions, and deletions in the DNA sequence. So mutations, what are the consequences of mutation? Mutations are basically a source of genetic diversity in population, and as already talked about, they can have a widely varying individual effects. In some cases, mutations prove beneficial to an organism by making it better able to adapt to environmental factors. In other situations, mutations are harmful to an organism. For instance, they might lead to increased susceptibility to illness or disease. In other circumstances, mutations are neutral, proving neither beneficial nor detrimental to the organism. Thus, it's safe to say that the ultimate effects of mutations are also as widely varied as the mutation itself. So we'll stop here. These are the types of mutations that we have discussed in today's class. So according to the question, the way I interpreted was that some mutations might lead to interference of the normal transcription process, but not all mutations will interfere with the normal transcription process. So that's why this particular statement is not correct, it's false. So we'll stop here for today's session. It's already 8. So if anybody wants to say anything, we can talk about that, or we can conclude the meeting for today and we'll meet again next Friday. Don't forget to submit the assignments. I think they have a deadline of 8th March or something. I have not checked. But anyway. So thank you everyone. If you don't have any further thing to say, we can actually conclude the meeting for today. Thank you. Does anybody has does anybody have anything to say? Okay, so we'll conclude the meeting today. Thank you for giving me 2 hours of a Friday night and interacting in the session. We'll meet next Friday, same link, same time. Thank you so much.